you from the standpoint of how are you interacting with the pathogens um, that we have started to discuss. Oh, come on. I clicked the wrong thing there. How are we starting to understand, or how are we going to understand these interactions? Okay. So learn some of the basic cells of the innate immune system. All right. Those of you that have had ANP2, a lot of this will be review for you, or some of the basics will be reviewed. Those of you that have had Bio2, you guys probably also touched on some of this, right? Understanding the mechanisms of innate immunity, right? And so, what are these cells doing to protect you? And then, even before the cells, we'll talk about mechanisms that your body has to try to neutralize pathogens. And one of the most important things that I start to try to point out here is that although we talk about the innate immune system and the adaptive immune system in separate lectures or units, I really am trying to emphasize how much these interact with one another. The communication between these two systems is essential. If you remove one of them, the other fails, right? They can't survive effectively, or they can't perform effectively without one another. So I'm going to try to drive home that appreciation. Oops. So, did I have it in here? Yes, I always forget this. So this is Eli Metnikov, who was Russian. He won the Nobel Prize in 1908. I always forget his name. And he essentially discovered the innate immune system. So this is a starfish larva here. And so you can see his little quote here. And so basically what he would do, he would take a little splinter, right, that you guys might get in your hand. He would poke it into the starfish larva, and he would notice that not too long after that, a bunch of cells would swarm around that little splinter that he put in. So he used a rose thorn, sorry here. And so that kind of led to the discovery or the speculation of the innate immune system, all right? You know, one of the ways that we can look at this is the innate immune system is a first responder, right? It's your body's first line of defense, all right? And so its main goal, honestly, is to prevent entry, right? Prevent that pathogen from even getting into your body, prevent you from having to detect it, prevent you from having to destroy it, right? But again, the innate immune system purpose, right? First line, hopefully nothing enters. Inevitably something will, and so once it enters, we have to be able to detect it. Once we detect it, we have to be able to destroy it, right? So that's the purpose of the innate immune system. You will see here as we go on, this third point, this destruction point, is where the adaptive immune system is going to play a major role down the road. All right. So from the textbook that I used to use, this was a description that they used, and I actually liked it a lot. And so basically, imagine yourself if you were in a military base, or if you were in a fort. If you like The Walking Dead on AMC, well, specifically, I like Fear the Walking Dead. I like that more, but that's just me, right? But this idea of, you know, they would set up a settlement and they're trying to keep something out. And so here you have your first line, right? You have your security walls, right? So that's meant to deter. Here you can see they have their barbed wire up. And so hopefully, right, that scares away the threat or prevents it from getting in. And so here you have your skin and mucous membranes. We'll talk about those in just a little bit, but they are incredibly vital. You also have, look, you guys have heard of these now, antimicrobial substances that different cells and glands of your body can secrete to help neutralize the pathogen, hopefully before it crosses one of these barriers, all right? And so one thing I want to point out here, right, when we say mucous membranes, remember, well, or don't remember, I don't know, maybe, but you probably talked about this in ANP2 or Bio2 if you've taken either one of those. Your digestive system is also called your alimentary canal. And although we don't think about it, it technically is an open system from mouth to anus, right? It's essentially one big tube that can be exposed to the outside. So the mucous membranes that make up your digestive tract are considered one of those first lines of defense, in case you were wondering that, because you're like, well, the mucous membranes are inside. Yes, they're inside, but they have a direct connection or access to the outside. And so they're considered a wall or a first line of defense. All right. Once a pathogen breaches your walls, which eventually happens, you then have to be able to detect them. All right. And one of the things that we use, and we'll talk about this, is something called pattern recognition receptors. So does anyone remember the receptor that is used to detect LPS? Does anyone remember that?
TLR4.
TLR4, right? Toll-like receptor 4. Toll-like receptor 4 is a specific pathogen recognition receptor, right? It is how you are detecting, right, that a pathogen has breached or gotten past one of your defenses or one of your walls, right? You also have something called the complement system, which you guys, I think, did as part of your assignment that was due this morning. And so we'll talk about that in more detail. But these are essentially a series of proteins that are in circulation that are looking for a target cell. You're going to go for a walk. Okay. Good luck. Love you, girls. All right. And so these proteins, right, have special abilities to recognize pathogens and activate the immune system. And they're just kind of looking for a target. We'll talk about those in just a little bit. All right. Once those targets have been identified, right, how do we get rid of them? All right. And you'll see an interferon response. And so these are cytokines, essentially, that your body can produce that can destroy cells and activate other cells of the immune system. Phagocytosis, that's a fancy word for eating. Once the complement has found something, right, it can be activated and do a whole slew of things that we'll talk about to the pathogen. Inflammatory response can help bring other immune cells to the area. And then a fever, right, which again, we won't focus a lot on inflammatory response and fever because we have so much to go through. But again, remember, pathogens are mesophiles. What temperature do mesophiles like to grow at a lot of times, Laura? Do you remember?
Is it body temperature?
It's body temperature, right? That's their optimal temperature. If we're talking about bacteria and potentially certain fungi, right? They like that 37-degree incubator, all right? So if we raise the temperature, sometimes that can help us eliminate them and interrupt their growth. Right? So here is your first line, right? Here's your skin. Also, right? Well, here, let's talk about your skin first. So those of you that may be familiar, your skin is made of something called stratified squamous cells, right? And here, this outermost layer, right, is basically dead cells and a lot of keratin, right? Remember, keratin is that protective protein that's made in endospores or put on the outside of endospores. It's also the protective protein that's on the outside of your skin. So not only do you have essentially this layer of keratin, you then have this stratified layer. And so this is protecting, right, or helping prevent bacteria from getting in, or a virus from getting in, or a fungus from getting in, right? Whatever pathogen you want to name, right, they have to get through all of this in order to gain entry in your body, right? It's very difficult. Now, you also have stratified squamous cells in your mouth, your esophagus, and in females, the vaginal canal. And the same basic idea, they are providing protection to that area of your body. And typically, pathogens are only going to get in if there is damage to this layer, right? Now, you don't have to think, this doesn't have to be like a big stab wound or something, right? There can be micro abrasions. This layer, truth be told, is actually relatively thin if you were to measure it. And so you could fall down and you could scrape your knee and maybe you think, oh, I didn't draw any blood, but you might see a little bit of skin that's pulled up, right? It doesn't take much, but as long as there's no abrasions in this layer, it's very protective. Here you see the mucous membrane, and we'll talk about this a little bit more, but essentially mucus is, right, sticky glue, right, that's secreted on top of these cells. And the pathogens get stuck in it, right? So basically our version of a capsule, right? Remember a capsule? We mentioned trying to maybe treat a bacteria with a certain drug or something, and the drug gets stuck in the capsule, right? It can't interact with the cell membrane, and so there's no infection. And the mucus does the same thing. And just to give you an idea of how good your body is at doing this, this is a cool immunofluorescent image of the small intestine. So here you can see the blue. This is a nuclear stain. This is staining epithelial cells within your small intestine. This is something called a fish stain. Don't worry about that specifically. It's basically staining bacterial DNA and making it fluoresce. And what you see here is that there's a clear divide between most of the bacteria in the gut and then the epithelial cells of your tissue. And this layer right here is where the mucus is. All right, so your body is really good at trying to keep bacteria away. Now, truth be told, right, these are not all bad bacteria. Some of these are good bacteria, but good bacteria, when given the opportunity to interact with epithelial cells, could do something bad. All right. Another important first line of defense are these microbes that are present. All right, and we'll actually talk about them in just a second, right? They are going to help keep the bad pathogens away. And again, just hold on to that thought. We'll get to it in just a second. All right. That first line of defense, right? We also mentioned antimicrobial substances. So on your skin, salt, right? We talked about some of those extremophiles that like salt when they grow. That's not necessarily, all right, the thing that regular bacteria or regular pathogens like. So if we keep the salt content high, we limit growth. Lysozyme, right, a natural, basically antibiotic, right? We talked about that briefly, interferes with the cell wall. Peroxidase, ooh, look, lactoferrin and transferrin, right? So this idea that you keep iron away, right? Defensins, which we just talked about. pH, right? So certain bacteria can lower the pH. I think you guys can start to appreciate that now. We mentioned that actually specifically in the vaginal canal of females. Lactobacillus keep the pH low, which helps protect the vaginal canal from pathogens. Other microbes, again, we'll talk about that in just a second. And then even the complement system, which we'll go over in more detail. All right. So let's talk about these microbial barriers, and then we'll take a short break. All right. So for these microbial barriers, all right, you have your normal flora, your normal microbiota, and that's totally fine. One of the major things that these guys or girls, I guess bacteria can be both in this case, are doing, right, is something called competitive exclusion. And it's nothing more complicated than what I have here, right? Space and resources, right? Let's say you live on a street and you don't want anybody to live next to you or across from you. One of the things you can do, right? And again, assuming maybe you have a lot of money, but you can buy the lot beside you. You can buy the lot across the street from you. And that means you prevent other people from moving in or other people from building. And the same idea here, right? Space. Just by being there, they are occupying a space that a bad pathogen may want to take. All right. Resources, right? Again, let's say I see Amanda and Allie, right, are trapped in a building together and it's an apocalypse and there's a limited amount of food, and Amanda decides that it's more important for her to survive over Allie. And so what does Amanda do? She hoards all the food to herself. Allie can't eat, and inevitably, sorry, Allie doesn't make it. All right. And the same idea here, by taking resources, right, and eating all of that, in this case, we'll say glucose, eating that glucose up, well, you don't leave any for the bad bacteria to get, and then they can't survive. Okay. And so, this simple concept is incredibly powerful. And this is what happens. I mentioned C. diff. Right? This is what happens. When you take those antibiotics, they basically wipe away all that bacteria, and then the Clostridium difficile has the opportunity to move in and take root, right? I've mentioned this before, but bacteria are a great source of antibiotics. They produce things or substances that help kill other bacteria, all right? Um, so Propionibacterium, E. coli, Lactobacillus, some of them are producing toxins that target or kill other bacteria in the area, right? So it helps control the bacterial population. So the bacteria in your microbiome are policing the other bacteria. They are helping to regulate what is present, all right? So really kind of cool relationship that we have with these bacteria. Right? And we'll talk about barriers being breached here in just a little bit. So I'm going to take a short break. It's 9:12-ish. Let's come back at 9:20-ish since we went a little long for the first part of class, and we'll keep on chugging through the innate immune system. All right. So this just kind of gives you a basic idea of the lineage of the cells that are involved in innate immunity, and I'll also just touch on the ones that are involved in adaptive immunity really quick. No matter what type of immune cell we're talking about, right, we have a common origin that you can see right here in the hematopoietic stem cell, all right? And so hematopoietic stem cell, right, is undifferentiated. It's waiting to receive a signal that tells it what it's going to become later down the road. And so here you have two kind of common or the two branches that are taken for the immune system are either the common myeloid progenitor, which is where we're going to see our innate immune cells come from, or the lymphoid progenitor, which is where our adaptive immune cells are coming from. And so what we're going to focus on over here is the left side of this chart. And so here, right, this common myeloid progenitor, right, does give rise to things that are part of your circulatory system via blood, right, and platelets that you find there. But what we will focus on today are going to be these granulocytes, which consist of eosinophils, basophils, and neutrophils. Also monocytes, which can differentiate into macrophages and dendritic cells. And here, importantly, what it's trying to show you at the bottom is that these macrophages and dendritic cells have an interaction here with the adaptive immune system, right? And so this is, right, where the adaptive and innate immune system are linked together is via macrophages and dendritic cells, right? But we're going to focus on these cells right here, and we'll mention on and off again the adaptive immune cells. All right. So neutrophils, all right? Neutrophils, basophils, and eosinophils, right? These are those granulocytes that we talked about before. We'll talk about each of them in a little more detail. But neutrophils are also capable of phagocytosis in addition to their granules that they contain that can have immune effects. Basophils, inflammation and histamine, parasites response. So if you guys have any type of allergies, these are actually two of the cells that play an important role in the allergic reaction. They do other things, we'll talk about that too, but this is where we probably become most concerned about them in our daily lives. Monocytes, right, are that other myeloid progenitor cell derivative, right? And they differentiate into macrophages and dendritic cells, which are phagocytes. And also we consider the dendritic cell the best APC, which is antigen presenting cell. Macrophages can also do this. We'll talk about the importance of being able to present antigen in relationship to the adaptive immune system, which is the lymphocytes that you see down here. We'll talk about those more likely tomorrow. All right. So for the granulocytes, right, I have a little bit of grenades here to show that A, they're powerful, and B, sometimes associated with these cells, there's a little bit of collateral damage, right? They're kind of that first responder once your um, defenses, right? The skin and the mucosal membrane have been breached. And so a lot of times with them, there's also collateral damage that's associated. So for neutrophils, these consist of 50% of all your circulating white blood cells. The majority of your white blood cells are going to be neutrophils, right? They're the first responder, or often the first responder to areas of infection. Basophils, the most important thing they're doing is releasing histamine. Uh, Histamine, when we think about allergies, is what contributes to our itchiness, our redness, perhaps some swelling, some runny nose, right? All that unpleasant stuff. But histamine is actually really important because histamine causes vasodilation. And when you have vasodilation, it's going to allow cells of your immune system to leave circulation and go to the site of infection, right? So it helps bring a lot of immune cells to the area that they're needed. Eosinophils primarily are going to be used for parasitic defense. And so those of you that have maybe been to third world countries or tropical places, sometimes outside of the United States, and maybe you've acquired a parasite, it's not abnormal. A doctor could take your blood and see that your eosinophil count is way up, and that might be an indication that you have a parasitic infection. Interestingly enough, too, any of you that have asthma, we've shown that eosinophils are present in inflamed airways in a lot of patients with asthma. They produce something called histamine ACE, which is meant to help regulate the histamine that's produced by basophils. Okay. Monocytes, right, I mentioned, can differentiate into macrophages or dendritic cells. Macrophages are kind of considered sentinels of the immune system. And so sentinel is a guard, right? They are interacting oftentimes directly with a pathogen, trying to eliminate it. Macrophages can be tissue specific. So microglia is a macrophage of the nervous system. If you've heard of Kupffer cells, these are macrophages that exist within the liver. Depending on the organ system and the tissue, they might not have a specific name, but they usually do have macrophages that are helping patrol that tissue in case there is some type of infection or damage to it. Dendritic cells, we consider these the scouts of the immune system. They are not necessarily interested in neutralizing pathogens. What they are interested in doing is gathering information about pathogens that are in the body and reporting that information back to other cells of the adaptive immune system. Okay. So this is actually a sentinel from a movie called The Matrix, which I learned last time I taught microbiology. No one in my class had seen. So I'm not even going to ask because I don't want to be disappointed. I couldn't get over that heartbreak again. All right. So for dendritic cells and macrophages, we consider these professional APCs, antigen presenting cells. Right. The big difference between these two, right, is not only is the macrophage presenting antigen, but it's interested in trying to eliminate or eliminate or remove the pathogen. Dendritic cells, right, they're just sampling and reporting back. All right. What these cells do is they digest the phagocytized material. We'll talk about this in a little more detail coming up. Essentially, though, they break them down with a series of enzymes and acids into little bits and pieces. They present those little bits and pieces on receptors on their cell, and other cells of the immune system can look at those bits and pieces and determine if a response is needed or warranted. And again, we'll go into that in more detail. This is a macrophage actually reaching out to grab bacteria. These are called pseudopodia. They're little extensions of the cytoplasm. They grab these bacteria and they pull it in to the cell where it can be phagocytized, eaten, and then broken down and presented on the surface. All right. Cool. Again, dendritic cells and macrophages, they are communicating with the cells of the adaptive immune system, which we'll talk about more in the next unit. Let's see. I wonder if this works again. Oh, come on. This was this was supposed to link to a video. Any of you that watch The Office, it's an episode where they're trying to teach Dwight how to actively listen for communication. And if you know The Office, of course, Dwight's not good at it. So they've like taken that video down multiple times. I'm still going to try to find it. Maybe we'll watch it tomorrow if I can find one. But how is, right, the innate immune system communicating? Communication is essential to the innate immune system being able to determine what is a pathogen and what's part of the host. And then it's essential for the innate immune system being able to call for help, all right, and get to the area of the infection. So that's what's going on here. Identification, all right, calling for help, and then getting to the area of infection. So receptors, all right, receptors are going to be transmembrane proteins. Again, they're spanning from the outside of the cell to the inside of the cell. They're going to be ligand specific. So they are looking for specific components. In this case, if we're talking about the innate immune system, they're looking for specific parts of the pathogen. Remember, those are called PAMPs, pathogen-associated molecular patterns, right? So they are looking for a very specific part of a, in this case, bacteria, but they can also do this for a virus and even certain fungal products, although we focus on bacteria a lot. Once that ligand, or in this case, that PAMP has bound to this receptor, some type of response is generated, right? This could activate the cell to make it more effective or efficient at removing the pathogen. It could tell that cell to send signals out to recruit other cells, right? There's a couple different things that can go on there, all right? So once that receptor has been bound, right, one of the things that can be produced is cytokines. The old textbook that we used to use called these the voice of the cell. So in this case, you have chemokines. We've talked about chemotaxis, right? So chemokine is a chemical signal that's helping direct a cell where to go. You have colony stimulating factors. These are going to help activate cells, perhaps turning a monocyte into either a macrophage or a dendritic cell, all right? So getting that cell ready to do its job. You also have interferons and interleukins. And tumor necrosis factor. These are inflammatory or can be inflammatory molecules. These can be molecules that activate other cells of the immune system, right? So they are their voice, their communication. When a ligand binds to that receptor, right, this helps activate the immune system via these cytokines. Adhesion, I'm not going to take the time in class right now for you guys to watch this video, but you can go and click on it. It's a really cool process. Where you actually see cells leaving circulation and entering tissue. One of the things you don't appreciate is that your cells are moving through your circulatory system very, very quickly. And so adhesion molecules have to be expressed on the endothelial cells of your veins and on the cell surface of your innate immune cells that are trying to leave, or even your adaptive immune cells that are trying to leave. And those receptors expressed on the endothelial cells and the cells allow them to stick, and then they leave the cell via a process called diapedesis. All right, this is necessary for cells to leave circulation and go to the tissue where damage has occurred. All right, so I'd go back and watch that video if you have time. It's actually pretty cool. So for some of these receptors, right, they're relying on this idea of pattern recognition. So pattern recognition receptors are looking for pathogen-associated molecular patterns. And, you know, one of the things I want you to appreciate is that conceptually, this is a relatively simple process. So when you see this, what type of butterfly is this? Does anyone know? Or what do you think it is?
Monarch?
A monarch, right? So it looks like a monarch. All right, good, because you kind of recognize that basic pattern, right? You've seen that before. Allie, if you saw this snake outside, would you pick it up?
Probably not.
Probably not. And why wouldn't you do that?
Because of the colors on it.
Because of the colors, right? And it's fine too if you said you didn't like snakes. Even if a snake wasn't venomous, I'm typically not looking for an opportunity to pick it up. But this is a coral snake, right? If you've heard the saying, red and yellow, you're a dead fellow, all right? And so these are patterns that we see, we know, and we recognize. Let's see. So here's another example. We'll go with Olivia. If you saw this pattern, what would you do?
I would stop.
You would stop, right? And so if you think about it, every time you pull up to an intersection, you don't take the time to be like S-T-O-P, stop, right? You're not like sounding that out. Your body recognizes that pattern. These pattern recognition receptors in your cell are going to recognize these same patterns. And what we see, and I just want to start to bring this up, is that we have things in nature that actually mimic these patterns to kind of trick us. And so here, right, this is something called the Viceroy butterfly, and it actually has a similar coloration and pattern to the monarch. I wouldn't be able to tell them apart. Shoot, what is this snake called? Well, anyway, red and black, you're okay, Jack. What is it?
King snake.
King snake. There we go, king snake. And so it's actually not venomous, right? But it has basically taken a similar pattern, and I'm not probably going to take the time to be like, oh, this one's okay to pick up. And so we see that in nature. Ooh, what is this a symbol for?
The NBA.
The NBA, right? And you guys, I could probably put other logos up there, and so you recognize patterns. And then I like this point too, because we'll actually talk about this a little bit more. One of the things that pathogens will do is they will try to mimic host molecules to hide or potentially protect themselves. Okay, but again, you guys know patterns, your cells know patterns. And this is a nice little diagram that shows, right, some of these important pattern recognition receptors. One of the things I want to point out is this pattern recognition receptors can be embedded within the cytoplasmic membrane of your cell, right, or your cell membrane, and they can also be in the cytoplasm. And so this is kind of nice because it means it can recognize things outside of the cell. It can also recognize if something's gotten into the cell, right? So here, these are toll-like receptors. These are important groups or important set of pattern recognition receptors. You can see that they recognize different things, right? So lipoprotein, they can also recognize LPS, although TLR4 is mainly what's used to recognize LPS. Here's TLR5 that recognizes flagellin from bacteria. Here you can see mycoplasmal lipoprotein, peptidoglycan, some other components of bacteria and fungi, right? Here's one even for protozoa, and these are all on or embedded within the cell membrane of cells in your body. Here, you can even see internally, you have TLR 3, 7, 8, and 9, recognizing viral DNA, viral RNA, and even bacterial DNA. You have these things called NOD receptors, which are also recognizing things in the cytoplasm. These are actually looking at components of the cell wall of bacteria, all right? So pattern recognition receptors, such as the one listed here, recognize pathogen-associated molecular patterns, right? Please keep those straight. I don't expect you guys to memorize this chart, but this is kind of a cool little signaling mechanism. If you remember when we talked about communication, right? And I said, hey, when a ligand binds to a receptor, it induces some type of effect. If you want to know how that happens, this is actually a diagram showing. So in this case, if a ligand binds to one of these receptors, it activates a series of proteins that are within the cell. What they both end up leading to is the translocation of a transcription factor, NF-kappa B, which is outside of the nucleus, and it goes into the nucleus. So basically, when something activates one of these receptors, it allows NF-kappa B to move into the nucleus, where it acts as a transcription factor for inflammatory cytokines, or in this case, interferon beta, which is important for communication between cells, right? So you can kind of see, what does that process look like? What does Professor Daft mean when something is activated? And this kind of shows you what's going on there. Again, within the cytoplasm, you have, let's see, okay, good. I couldn't remember if I had another one after this. You have your nod-like receptors and your rig-like receptors, and they are basically going to activate and form something called the inflammasome, which is where you have multiple organelles that come together and destroy pathogens that are in the cell, right? But these nod-like receptors and rig-like receptors are focused on pathogens or components of pathogens that have managed to enter the cytoplasm, all right? Whereas toll-like receptors can be cytoplasmic, right? Or they can be embedded within the cell membrane to detect what's going on outside of the cell, okay? So one of the things that I previously mentioned was this idea of NF-kappa B. So when we talk about activation of the innate immune system and even activation of the adaptive immune system, NF-kappa B is the major transcription factor that's driving that process. Interestingly enough, NF-kappa B is an important transcription factor in a lot of cancer cells. And although we know this, it's not really a good target because it plays such a big role throughout our whole body. And so if you were to target it, you would have a lot of major side effects. All right. NF-kappa B stands for nuclear factor kappa light chain enhancer of activated B cells, right? So this is what was discovered in, all right? Don't worry about memorizing that name, but in case you want to know what NF-kappa B stands for, that's it. So it's essential for the regulation of immune responses. I already kind of pointed that out, but this is the basic premise of how it works. NF-kappa B has a protein attached to it called I-kappa B, inhibitor of kappa B, all right? As long as this inhibitor is bound, NF-kappa B cannot enter the nucleus where it acts as a transcription factor. Okay. So here's my boy Nemo, all right? Ooh, this is Nemo from, how many leagues under the sea? Yeah, there he is. He's there, right? But NEMO stands for NF-kappa B essential modifier. And so NEMO, right, binds to, right, NEMO represented here by the beard, binds to I-kappa B. And when I-kappa B has NEMO bound to it, it detaches from NF-kappa B. NF-kappa B is then able to enter the nucleus where it acts as a transcription factor, right, for cytokines and other proteins that are associated with the activation of the innate immune system. Right? Does that make sense? Right, kind of basic premise of how NF-kappa B is working. Okay, now the complement system. I'm not going to spend a lot of time on this. I want to go over the basics. This is a link here that I think is a Khan Academy video that goes into a little more detail. But one of the most important things to realize about the cascade phenomenon, or not the complement, one of the most important things to realize about the complement system is it starts something called a cascade phenomenon. And so you basically have activation of these different complement components start feeding off of each other and lead to the activation of the whole complement system. So it works quickly and it is a very strong response. For the complement system, and hopefully you guys recognize some of these things from the questions that you worked on that were due this morning. You have the classical pathway, right? This is antibody antigen dependent. Your next two pathways, the alternative pathway and the lectin pathway, are antibody antigen independent, right? So the classical pathway, you're going to need the presence of antibodies, and these antibodies, right, are actually produced by the adaptive immune system, right? So another link between innate and adaptive. So this is a diagram from the textbook that I previously used. I actually like it. It helps kind of simplify this. It says, here's the three pathways, alternative, lectin, classic. Here you see that the classic pathway is antibody dependent. The alternative pathway is not antibody dependent. No matter what pathway you start with, you form an enzyme called C3 convertase. C3 convertase then splits complement protein 3. Complement protein 3, right, becomes C3a and C3b after the split. You see here C3b is important for coding pathogens. So C3b, you can see it all the way up here for the alternative pathway, codes pathogens. Your phagocytes, macrophages, and dendritic cells have receptors for C3b. So if they see C3b, they know that it's coding a pathogen. They can engulf it. C3A, right, is important for raising vascular permeability, all right, and acting as a chemoattractant. C3 convertase, right, combines with C3 to form an enzyme then that can also split C5, right, complement protein 5, C5a along with C3a, right, is increasing vascular permeability and acting as a chemoattractant, while C5b forms an important component of something called the membrane attack complex, right? And so this is punching a hole in the cell membrane of the target cell, which makes it impossible for it to maintain homeostasis, which leads to its death. Antimicrobial peptides like defensins are doing something somewhat similar, right, by disrupting the membrane. So the big takeaways from the complement system are, what are the three pathways, right? What kind of starts them off? And then this idea that they all convert on the formation of C3 convertase. And then once you form C3 convertase, right, through the cleavage of C3 and C5a down the road, you have three basic responses: the inflammatory response, right, vascular permeability, chemoattraction, you have the formation of the MAC complex, and then you have opsonization, right, another word for phagocytosis of the pathogen, right? So the basic three things that the complement system is doing, right? What are they? Good, doing good on time. So let's talk about phagocytosis, right? This ingestion of a pathogen, by the immune system. So you have six major things that are going on: recruitment, recognition, engulfment, maturation, digestion, and exocytosis. Right? I know that's quick, but we're going to talk about each of these points together. So recruitment, right? Chemotaxis, right? This is different from bacteria in the sense of what's attracting these cells, right, is not going to be nutrients like we see in chemotaxis for bacteria. So here we have chemokines, right, phospholipids, and some not phospholipids, that should be phospholipids from bacteria, other bacterial products, and C5a from the complement system, right, are really important for chemotaxis, right, helping draw those macrophages and those dendritic cells to a site of infection, all right? Not surprisingly, once you find the site of infection, right, you have to recognize it. You have to be able to find it once you get there. So we use those receptors that we talked about. Perhaps pathogen recognition receptors can provide further activation, or remember in the previous picture, receptors for C3b, which are hopefully coated around pathogens, will let the cell know what's going on. So in this case, opposins, right, are an indirect recognition. So that would be like C3b bound, right? The phagocyte has a receptor for C3b, and so that's how it recognizes a pathogen. Some phagocytes can recognize mannose, which is a sugar on the surface of bacteria, directly, and just pull that bacteria in with just that recognition. Engulfment, you guys saw previously in a picture, pseudopodia. This can help surround and bring cells in, and then they'll enter via endocytosis. Maturation, basically when a phagocyte is getting ready to consume something, we start to produce more antimicrobial, we activate the antimicrobial capabilities of that cell, all right? And so we then fuse it with a lysosome, which contains enzymes and acids to help break down and destroy that pathogen. That starts the digestion process. Once it's digested, we expel most of that pathogen, right? So this is basically phagocyte poop. But some antigens are held onto and presented on the surface of the cell to be looked at by the adaptive immune system. I realize that may be a lot. And you're like, what does this all mean? This is actually a visual representation excuse me, of what's going on. So here you have chemotaxis. In this case, it's showing that C5a attracted a phagocyte, but it could be other components of the microbe. Here you have recognition and attachment. So in this case, it's showing that pseudopodia with C3b receptors reached out to bind onto C3b. So it is then engulfed via endocytosis, right? When it's taken in via endocytosis, this is something called a phagosome. So here you see the pathogen that's engulfed by the phagosome inside the cell. You then have fusion with lysozyme. So this forms something called a phagolysosome. And this phagolysosome, right, breaks down the pathogen. Here it shows exocytosis, so we expel that pathogen. What it's not showing is that some of this pathogen can be presented on the surface of this cell for potential recognition by the adaptive immune system. Okay. So for macrophages, which are going to be one of your main phagocytes, these cells can be inactive for weeks or months, right? So they basically are on patrol looking for something to recognize, right? And that might not seem like a lot to you, but days to weeks is actually a relatively long lifespan for your cells, right? You guys turn over, even your healthy cells are turned over on a regular basis, right? As I mentioned before, they can be or they can take residence in tissue. And so they can hang out in specific tissue waiting for some type of recognition, right? And this is where we give them some different names, and they even possess slightly different attributes depending on the tissue they're hanging out in. They have multiple activation states, right? So here they have an inflammatory activation state. Here they have a suppressor activation state. Why would you want a macrophage that has the ability to suppress? Why would that be good, Anna?
Sorry, what? You're abstract.
That's okay. Why would you want a macrophage that is able to induce inflammation? Well, we kind of know that, but why would you want a macrophage that's able to suppress inflammation?
So that, sorry, my puppy's distracting me.
That's okay.
Suppressor. I mean, so that it can kind of do the opposite, like.
Right. I mean, I'll take that. Right. Essentially, you want to be able to stop the inflammatory response. So when your body responds to a pathogen, oftentimes it needs signals that help it stop. If you know people that have autoimmune diseases, in some cases, their ability to stop inflammatory events is limited or hampered. And that's what leads to them having that autoimmune disorder, right? Professor Perry actually mentioned giant cells when she was talking about some fungi in the lungs, all right? And so basically, macrophages will try to ingest and destroy certain pathogens. Sometimes they can't, and so what they do is they essentially swell up and take in as much pathogen as they can, and then other cells in your body surround them, and that's where you get some of these granulomas, okay? And I can't remember, some group is doing TB for the disease project. I can't remember who it is off the top of my head, but they'll be talking to us about granulomas, I would assume, in the not so distant future. All right. Neutrophils, which are also capable of phagocytosis, have a very short lifespan, right? They're the first responder. This is the highest turnover of cells in your immune system. They also can produce these cool things called nets. And so basically what they do is they cough up their DNA, which is really, really sticky, and embedded within that DNA, they have proteins that are toxic to the pathogens, all right? And so here you can see a neutrophil that has released this net. Bacteria is trapped in it and it ends up killing that bacteria. Unfortunately, this is a terminal event for this neutrophil, right? it can't suck its DNA back in and go on about its life. All right, so this is kind of its last hurrah. But this is possible because, again, there are a lot of neutrophils and we turn them over very quickly. And so it's not as big a deal to lose them to an event like this. All right. Dendritic cells, another type of phagocyte, right, have a very similar lifespan to macrophages, days to weeks. Dendritic cells are circulating. Remember, they're not typically going to take up residence in tissue because they're scouts. They're just trying to find whatever information they can, report back to the adaptive immune system, so they're kind of always on the move. Dendritic cells are one of the most important bridges between the innate and the adaptive immune system. Don't worry about these two types very much of dendritic cells, myeloid and plasmacytoid dendritic cells. But what I do want you to realize is that these myeloid dendritic cells are important or essential for the activation of T cells in the adaptive immune system, all right? Part of the ability to activate means that you're great at producing cytokines. As the cytokines that are produced by dendritic cells are essential for activating cells of both the innate and the adaptive immune system. And remember, if we're talking about an antigen-presenting cell, right, a cell that is great at doing its job, that would be a dendritic cell. But please don't discount macrophages. They can also present antigen, but they have other jobs and functions too. All right, cool. We made it through the innate immune system. So what I'm going to do here, all right, is give us, it's 9:58. Let's just move in, if you guys are okay. Let's move into the lab portion of class, and we can start with our presentations, and then you guys can get a jump on your assignments today, so that we're not maybe working quite as late as we were yesterday. Okay, so I'm gonna go ahead and